Lichens are organisms that were traditionally thought to be comprised of two separate organisms that live together as a collective. One organism was believed to be a fungus or the microbiont, while the other one was an alga or cyanobacterium, otherwise called the photobiont. The fungus organism of a lichen was thought to be restricted to the phylum Ascomycota, and is the dominant organism of the relationship. The alga or cyanobacterium is a unicellular photosynthetic bacterium. The two organisms were considered to be in a symbiotic relationship, since both organisms directly benefited from the partnership. The symbiotic nature of the alga/cyanobacterium and the fungus was first discovered by Swiss botanist Simon Schwendener in 1867. The relationship is symbiotic because the microbiont benefits by gaining a carbohydrate energy source via the photobiont, while the photobiont benefits from gaining a shield from harmful UV light.
Because lichens have been studied for over a decade, they are thought to be well understood on the basic level. The idea of lichens being comprised of a fungus and cyanobacteria or alga working in unison has been accepted without question. However, despite lichens having been studied for so long, certain characteristics were not understood. For example, when the fungus and cyanobacteria or alga known to form a lichen were combined in a lab, the synthetic lichen did not produce anything that resembled the natural lichen. Another phenomenon that perplexed scientists was the fact that two lichen species analyzed within this study, Bryoria tortuosa and Bryoria fremonti, are comprised of the same fungus and algae. B. tortuosa is yellow in color and produces vulpinic acid, a toxic substance, while B. fremonti is brown and doesn't produce this acid. Even though these lichens seem genetically identical, as no difference was found in the fungus and algae that form it, scientists could not explain the phenotypic differences. The authors initially hypothesized that the differences in phenotype were due to varying gene expression between the two species. However, an analysis of gene expression of the two species revealed that this was not the case. When the gene expression analysis was expanded to include all fungi, it was revealed that another type of previously unaccounted for fungus was present, Basidiomycota. This finding flips everything we previously knew about lichens on its head. Rather than a lichen being comprised of just one fungus and a cyanobacteria or alga, there is in fact a third partner found to be involved in many different types of lichens, Basidiomycete yeast. This finding explains the phenotypic differences in the lichens mentioned earlier. B. tortuosa contains high amounts of yeast, which results in its bright color and toxic substance produced, which helps protect against microbe invasion.
For years, scientists have been using lichen as a major symbol for symbiosis. The article mentions that lichen was one of the first organisms to inhabit land, and this is because of symbiosis that makes this organism so successful. In Module 1 of this class, we discussed how the earliest fossils appear to be cyanobacteria, which play an important role in the lichen relationship. This also supports the claim that lichen were likely one of the first land-dwelling organisms. In order for species to survive, there has to be dependence on other organisms for resources, like food or shelter. Since the beginning of eukaryotic cells, there has been evidence of symbiosis. Eukaryotes are descendants of a chimeric organism that was a combination of a host cell and the cells of an alpha proterobacterium that found shelter inside of it. This major theme of symbiosis in the origin of eukaryotes is known as endosymbiosis. This happens when one cell engulfs another, such that the engulfed cell survives and both cells benefit. After multiple generations, a symbiotic relationship can result in two organisms that depend on each other so that neither could survive on its own. On the website, this was explained using eukaryotic cells and mitochondria. The idea of organisms becoming dependent on one another for survival is what is hypothesized to happen in today's lichen.
Various pieces of evidence support the assertion of yeasts being embedded in the cortex of lichens. Recent research has involved the investigation of lichen-inhabiting taxa within Puccinio-Mycetina using molecular data. The subphylum Puccinio-Mycetina includes a large number of unicellular and dimorphic fungi, which are usually studied as yeasts. Dr. Spirabilli and John McCutcheon, a professor of microbiology at the University of Montana, found that a previously unknown second fungus is a third species that forms lichens. This is the Basidiomycete yeast, and it appears to form the outermost crust of the lichen organism. To determine whether a certain uncharacterized Basidiomycete was to be found in lichens other than the studied Bryoria species, primers for rDNA were designed to screen lichens growing next to Bryoria in Montana forests. Each species carried a genetically distinct strain of Basidiomycete, indicating high specificity. Furthermore, another common lichen species, Lotharia vulpina, carried a Basidiomycete genotype distinct from those in Bryoria not only in Montana, but also in northern Europe. Thus, Basidiomycete was assayed across the 7 main radiations of macrolichens in the class Licheneromycetes, and related Basidiomycete lineages associated with 52 lichen genera from 6 continents were found. This data indicates that Basidiomycete fungi, or yeast, are ubiquitous and global associates of the world's most speciose radiation of macrolichens. Another study conducted in the South Shetland Islands of Antarctica stated that Antarctic terrestrial ecosystems are largely dominated by lichens. The study intended to isolate and evaluate the diversity of yeast in different species of both macroalgae and lichens collected in the islands. 200 of the 405 yeasts collected were from lichens, with 18 variations of lichens being identified. The study was the first to report yeast communities from the Antarctic lichen Ramalina terebrata. The results suggest that Antarctic lichen sphere represents a huge substrate for cold-adapted yeasts.
DNA analysis suggests that yeasts have been embedded in the lichen cortexes since the end of the early Cretaceous period, and the discovery of this symbiosis, according to Biol, seriously challenges a lot of assumptions that have been held by lichenologists for a century. The symbiosis that occurs in lichens proves to be one of the oldest and most successful symbioses in nature. They are truly a diverse and evolutionary critical relationship. Going forward from this finding, our next point of research would to investigate whether Basidiomycete yeast play a role in the structure of lichens. Currently, synthesizing lichens in lab has had fruitless results, but by expanding on the functions of these newly found yeasts, scientists may come a little closer to creating lichens in lab and thus being able to study them without having to collect them in nature. Ultimately, this new finding on the makeup of lichens applies on a broader scope to our understanding of the symbiotic world. It depicts how multifaceted symbiosis can be and how the facts that we may think are set in stone are prone to variability.